one standardized short DNA sequence for species identification. And so we will use this sequence, and therefore we have to choose a part of the genome that actually evolves quickly enough to access recent speciation events. So we will then focus on mitochondrial DNA because this evolves more quickly than nuclear one. And as a standard barcode marker for animal taxa, CO1, a protein coding mitochondrial gene, has been designated. And for this species, it actually works like for a typical barcode. So, for example, when you go shopping, you scan the specimen, and by using the barcode, you can actually find out to which species it belongs. So then we actually compare the DNA sequence, which is quite short, so like around 600 base pairs, and then we try to find whether there are differences. The more differences there are between specimens from a certain level, we will call them different species. Simply explained. So we have the specimen, we process it, and then finally we obtain a barcode, which we will try to match in a barcode library, which has been previously established with sequences of known identity. But there is, it's not so simple, and there is one major condition which we have to have for that DNA barcoding works efficiently, and that is a distribution of intraspecific variation versus interspecific divergences. So in red we have the intraspecific variation, which has to be much lower than the interspecific variation, and so the ideal case is represented here, and there should be this bimodal distribution of intraspecific. In red should be, and there should be a barcode gap between the highest interspecific divergence and the lowest interspecific divergence. And because in many animal taxa you have a much lower interspecific variation, often one order of magnitude lower than interspecific divergences, we can actually differentiate species. And this has worked and not worked. For example, on some deep sea taxa such as corals, it didn't work because divergences are too low. But on amphipods, CO1 is actually a good barcode marker, which I will explain later as well. So the studies I will present, I have conducted on Lisanacerid amphipods. Why? Because they are a good model group for several questions. Because they thrive in cold water, so they are very abundant and diverse in shallow waters, in polar regions, as well as in the deep sea. And there they are abundant and diverse, and they are mostly scavengers, so they have lots of life-size pelagic species. The first case study is a DNA barcoding approach on the complex of genera Orthogenic, so the key species, and I have used two genes and a reverse taxonomy approach. The second case study is the one on Muretan and Scylus. Actually, this does not fit in this workshop because it's megafauna, but since the results are interesting, I decided to present them anyway. And for this, I have used phylogeographic tools and two genes and as well a reverse taxonomy. So how do we sample these species? You can sample them with the Agassiz straw and the Rorschach dredge. The Rorschach dredge is a small dredge you can see below the Agassiz, the huge Agassiz straw, and which has a kind of, it samples smaller sized fauna, and the EBS, as we talked before, and as well the amphipod traps. So these are baited traps which are attached to a lander system you can see here. And then bait, so that fish are then put in this kind of cages you see below, and they have a narrower, so bigger entrance so that specimens can easily enter, but they have difficulties coming out, so they are easily caught. The system works like this. So because of the weight, the lander sinks down, and then it stays on the bottom for 1 day or two, and then you come back, you release the acoustic signal, and this makes that with the acoustic signal that the releaser opens, releases the weight, and because of the floaters in orange, the whole system comes up again, and then you hope to find it again. And in these traps, you then find a high number of specimens per species, which allows you to easily conduct phylogeographic studies and population genetic studies. So for this, it is very interesting to really find out the answers to questions on connectivity. And the methodology I used is that I've always used 96% of ethanol or absolute ethanol, which had been pre-chilled in the cooler before when we put the specimens inside, and specimens are stored at -20 as well after collection. And I have extracted DNA using standard kits and PCR using universal primers and antigen-specific primers, and then standard cyanic sequencing. So the results on the first study I will present here. We have sequenced around 250 specimens belonging to 25 different species. And to see whether the approach is really efficient, we needed to plot and to find out the difference between the interspecific variation, which was an average around 0.3%, and in yellow, the interspecific variation, which was around 11.6% in average. So we did find a barcoding gap between the highest interspecific variation and the lowest interspecific divergence, which made the approach efficient to assign to our clades, so to our specimens, whether there are interspecific or intraspecific relationships, so we could see, we could assign the species to names. So also our morphospecies were always retrieved in monophyletic clusters, which is a sign that the approach works well. And because of this, we could also check whether the genetic clusters, which corresponded to undescribed species, whether they were morphologically different, and they were. So we found 8 new species, and 2 of them have been described so far. And also, we used a nuclear gene afterwards, and we could see that the topologies have been confirmed, because this is actually the major criticism on DNA barcoding, is that it's based on a single gene and then a mitochondrial one, so that there are some problems, for example, incomplete lineage sorting, which means that the gene tree does not always reflect the species tree. These problems could be excluded because the topologies were the same. And this approach also allowed us to test hypotheses on faunal distributions. So for some species, we found a genetic homogeneity between specimens over a large sampling site. So we could find divergences in red, so in the range of interspecific variation between specimens all around the Antarctic, in the South Atlantic and the North Atlantic, and over a bathymetric range of 300 to more than 4,500 meters. So this species is confirmed to be widespread and truly Europathic species. Europathic means that it covers a wide bathymetric region. The second species, Abyssopomene chiquix, we found the same. We found a very low genetic divergences between specimens from the North Atlantic, South Atlantic, and around the Antarctic continent. And over a range of 2,000 to almost 5,000 meters, so this is an exclusively abyssal species, and it is widespread. And on the other hand, in some species, we found very high genetic divergences, so we found several lineages, and between these specimens, we had divergences in the range of interspecific divergences between known taxa. So you can see that in colored stripes, you could see several lineages. And when we looked at these lineages, we could see that some of them were restricted to some localities, while others were more widespread. But again, I would like to underline that often when you increase sampling, you might find specimens, and these restricted range might become more widespread. So then we used these molecular results actually to have a look at the morphology in detail. So when there were cryptic species or lineages, we tried to see whether they're really cryptic, or if we could find differences, or we could sometimes synonymize these, or for example, one species that had been synonymized, we could separate them again. And we could also describe some species based on this, so we found this clade. And there were 2 genera mixed, and one genus, Abyssopomene, was scattered all over the tree. So we could revise it and then actually name one genus as a whole clade and describe 2 species. Then we also had extended DNA barcoding approach to other Lysianacerids, so not only this complex of genera, but a more wider range. And again, we found this bimodal distribution and this barcoding gap, which allowed us to apply the method and also find species complexes in 2 species, Eurystes murali and Valdeckia oblis. So again, for these clusters, for Eurystes, the genus Epimelia, Acrotopus, which is a typical deep sea genus, and then also for the deep sea Foxocephalids. Then my second study was on Meridiana spillis, so not a macrofauna species, a giant actually, and it is known as one of the most widespread species of all amphipods because it occurs from just below the surface to more than 7,000 meters and all around the world in every ocean. And for this species we found that this giant, actually very well studied species from terms of biology, it is actually not one species but composed of 9 species. And these clades had really, for each clade the distribution was different, and for example, we found 2 widespread clades, one bacterial, so above 2,000 meters, and one cosmopolitan abyssal clade, so below 2,000 meters. So the bipolar clade was actually the first genetic evidence for a larger species that had a bipolar distribution genetically confirmed. And the cosmopolitan was found in the Pacific, Atlantic, and Southern Ocean so far. Then in the Brazil abyssal basin, we found 2 clades occurring in sympatry, and another, we found one single specimen which was genetically divergent from all others at this seamount in the Pacific. We found an Atlantic clade at abyssal depths, which was found again in the Brazil abyssal basin. So in the same basin we found 3 species co-occurring, and this was genetically the same as one in the Gulf of Mexico. Then we also found an Atlantic bacterial clade, which had specimens from around Iceland and the Bahamas, and then an Antarctic abyssal clade, so grouping all specimens at abyssal depths around the Antarctic continent. And then finally, the last species occurred again on this seamount in the Pacific. So on this seamount in the Pacific, we also found 3 species co-occurring but at different depths. And this is another method to explore a phylogeographic method, which is a haplotype method. Actually, it groups, so a haplotype is a unique sequence and it represents a circle. So the bigger the circle is, the more specimens have the same haplotype. And when you have these black dots, means that the haplotypes are unsampled or they don't exist. But it means that when you have they are separated by several mutational steps, which means the different dots. So the more they are separated, at some point they break off because you put the threshold, and then actually they represent different species. So we found 2 species above 2,000 meters, 2 bacterial clades, and below we found 6 abyssal clades. So actually the abyss for this results appear to be more diverse than bacterial depths, which is often the opposite we find. And we also found this strange result, strange in the sense that we didn't find a single clade that grouped specimens from above and below 2,000 meters. So this 2,000 meters seemed to be something, a kind of a barrier for reproduction and gene flow. So when we compare species richness, actually this goes against the depth differentiation hypothesis, which is a hypothesis that states that genetic or morphological differentiation decreases with depth. So actually, in this case, we should find less differentiation at abyssal depths, where we found the opposite. And also, the bacterial groups seem to be more widespread. In abyssal, we had some lineages which were really restricted to single regions. But again, this might change when you increase sampling, you might find more. Again, in the Pacific, we found 3 species-level clades. And this genetic break at 3,000 meters is not, for Eurytinus, it's not the first time we find this because it has also been found for bivalve species. So bacteriometry plays an obvious role in speciation events. This is clear from this study as well as from many others. And to see whether these genes were good to differentiate species, yes, because for all 3 genes, even if they evolve at different levels, at different speeds, we found always a bimodal distribution of interspecific variation in red, the average values, and interspecific divergences in yellow. And for CO1, we actually found a barcoding gap, so there was no overlap. For 28S, there became an overlap because it's a very slow evolving gene. But still, we could more or less differentiate species based on this as well. So then we again used the reverse taxonomy then we also found a unique combination of morphological characters for every clade, and they are now currently being described. Then the second approach for which molecular tools can be used is the blind taxonomic studies, and there we will only use molecular operational taxonomic units. And it has been shown that they can be used for comparative diversity studies on abundant but understudied taxa, so for which we have very little knowledge. And especially on amphipods, actually, the basic thing for these kind of studies is that we have to find a threshold, and this threshold actually represents a cutoff value of divergence, where we say this is now a different species or not. And it's quite important to have this right, otherwise all conclusions can change. But since there have been a lot of barcoding studies using morphospecies and checking their divergences, we can actually base our thresholds on this. So there have been quite some studies on barcoding on amphipods. And this is what they did in the study of Knox et al., where they actually used only motors to check the abundance and diversity of oxocephalids, a very taxonomically difficult deep sea group. And they decided to use a threshold value of 6%. So above 6% of genetic divergence, you would consider this as different species or motifs. And then they compared and found that they had around 50 different operational units, and of which more than half occurred at only one locality. And they also found at several stations new molecular operational taxonomic units. So actually the same results as we would find with morphology, just a bit more easy. And so to conclude, DNA barcoding is efficient for amphipods because we always find this bimodal distribution and the presence of a barcoding gap, allowing these species assignments. And also we treat morphospecies as monophyletic clusters. So there is no, for example, one with the same name appearing in one clade and another, so these problems did not appear. And DNA barcoding is not only useful to identify species, but we can also really use it to test these hypotheses on fauna distribution, so to check whether species are endemic or Europacific cosmopolitan at a large scale, but we could also do it at a more smaller scale, but this was not the case for me. And then we can also use them on large scale comparative biodiversity studies, so also in the zones of interest for mining. And then it is now clear that for this anastoid, species richness is really underestimated because, I repeat again, this huge species Eurythena scalus is one of the icons of the deep sea fauna, and even for this species we found 8 overlooked species in one, which is crazy because it's big and everybody knows it. And so genes can also serve beyond species identifications. So you can actually test general hypotheses on deep sea ecology using molecular tools, and this can be crucial to understand better the deep sea ecosystems and their vulnerability to human-induced modifications. And one of the first things we should study is genetic connectivity, because depending on the extent of genetic connectivity, a population which has been affected could be actually supplied by migrants from an unaffected zone. So this is very important in this context. And this we could study using fast evolving markers, high resolution markers such as microsatellites and SNPs, single nucleotide polymorphisms, which before microsatellites were actually hard to isolate and characterize, but now with the use of next generation sequencing tools, this becomes much more easy. So it's feasible now. And then we could use F statistics and mobile network analysis and other analysis, which allow actually to test whether there have been population expansions or bottlenecks in the past, and this can serve to actually determine the vulnerability of a population and the possibility of recolonization after disturbance. Also, phylogenetic analysis using several genes together can elucidate more about the evolution of deep sea taxa, for example, and also to test, as you could see from the results before, general hypotheses in deep sea ecology. For example, the depth differentiation hypothesis, the source sink hypothesis, which actually states that abyssal populations are not viable, that they should be supplied by migrants from bacterial depths. And since we found this genetic lake at 3,000 liters, and it has been found before, and people find cryptic species at different depths, this hypothesis is not valid, so there is no possibility of gene flow between bacterial and abyssal depths. And this is very important, all these findings, in the light of conservation and management methods and decisions. So what we also found is that abyssal organisms appear to be low, it's not true, and populations do exhibit genetic structure. Thank you for your attention and for inviting me here at this nice group.
Questions? Stefan?
Yes, thank you. Has barcoding ever been used to compare two different sets of morphotypes? So if I think Gordon earlier on said that some animals have been maybe described as species A and in other studies as species 1 or whatever. Has it ever been tried to compare two data sets like these to see if they actually the same species?
I guess that when people just sample and they sequence and they compare it with the database on GenBank, they could already find whether their identification is okay. I don't know whether in this particular kind of study this has been done, but it is obvious that it is feasible and probably has been done.
I have a question. This is a very ignorant question. I see that you are using the 16S and 28S of the mitochondria, so that you need the light material. That's why the cooling and the preservation and so on, right? But you use ethanol as a fixing.
Yes.
Right.
Yeah.
So that means that all samples that have been preserved in ethanol can be barcoded again?
All samples and also in formalin, but then often DNA is degraded, so the sequences are much shorter. But there are now methods that allow retrieval of sequences from old museum materials, but it's harder. But with next generation sequencing tools, it becomes also more easier. So it's not excluded, but the better would be not to conserve specimens in formalin.
This is very important for us, for the Secretariat, because we have samples that they are as old as 10 years. And they are being reported to us as amphipola, but they give us a number in density. And I'm sure it's all the species that you are talking about, maybe 2 or 3 individuals of each of the species.